Hello, everyone, and welcome to today's live broadcast, Recent Trends in GPCR Drug Discovery, Novel Targets, New Disease Indications, and an Upsurge in Biologics, presented by Dr. Annette Gilchrist, an Associate Professor in Pharmaceutical Sciences at Midwestern University. I'm Susie Valdez, and I'll be your LabRoots moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I want to remind everyone that if you have any questions, please click on the Ask a Question box located on the far left of your screen and type the questions into the drop-down box that appears on the screen. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, please click on the Help Desk button located on the promotional board at the bottom center of your screen, or use that Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Just click on the accreditation button located on the promotional board at the bottom center of your screen and follow the process to obtain your credits. Please join me now in welcoming our presenter today, Dr. Annette Gilchrist. I will now turn the presentation over to her. Welcome, Dr. Gilchrist.
Thank you. I appreciate the invitation and the opportunity to talk about some of the recent trends that are occurring in GPCR drug discovery. GPCRs are a very well-studied target, and if you actually go to the slide, you can see that this superfamily is the largest receptor family. There are over 800 members to this family. They constitute 3% of the human genome, and they mediate a very diverse set of ligands, everything from photons to odors. For sensory signals, you can have taste. Those are all G-protein coupled receptors. Things like neurotransmitters and chemokines. There are a lot of peptide receptors, and recently there's been an interest because some of your bacterial metabolites from your gut have been found to activate G-protein coupled receptors. From those 800 or so receptors, about 127 of them still have what they refer to as orphan status, and that means that they actually don't know what the endogenous ligand is that activates that receptor. So typically what we do is we divide the mammalian GPCRs into 5 families. The first is the family A. That family is the largest family. There's almost 300 members to that family. Family B, which is the secretin family, has things like calcitonin and parathyroid receptor in it. We have a family known as adhesion receptors. That's family B1. You have family C, which is the glutamate receptors. For that family, it only has 22 members, but many of them are quite important. So the calcium sensing receptor, metabotropic receptors, the GABA receptors, those all belong to that family C. And finally, frizzled. The frizzled family has 11 members. There's been some controversy as to whether frizzleds are in fact G-protein coupled or not. However, I think that many of us believe that they certainly belong in the GPCR superfamily. So people have studied G-protein coupled receptors for many, many years, and that's because oftentimes they recognize that very specific point mutations in G-protein coupled receptors can lead to disease. So I've listed a few of those on this slide. You can see, for example, that if you have a mutation in your rhodopsin receptor, it can lead to retinitis pigmentosa or stationary night blindness. Oftentimes, these are either gain of function mutations, as is the case with the V2R receptor, vasopressin receptor, or loss of function mutations. But perhaps the most commonly known GPCR mutation is, in fact, red versus green. So in the object that you see on the left side, if you can see a red star and a red circle and a green number 6, you are not colorblind. If you can't, though, it probably means you are part of the 8% of the population at large that is in fact colorblind, and that is in fact due to a mutation in a G protein coupled receptor. So people have studied G protein coupled receptors in terms of drug discovery for many years. Historically, they represent nearly 34% of all the drugs that are on the market today. In fact, I would strongly recommend a recent paper that's come out in Nature Reviews Drug Discovery that, in fact, discussed the 108 different targets, meaning 108 different G protein coupled receptors that have drugs for them, and that's what this graph is on the right. And so if a receptor has been targeted by a drug, it has a red circle, and the larger the circle, the more drugs that are available for that specific target. These 108 receptors account for 475 approved drugs. Of those, nearly 70, so 69 of them, have been approved in just the last 5 years. Now, I will point out that there is a slight discrepancy between two recent reports, between the Nature Reviews Drug Discovery and a Mulpharm paper from Paul Inzel's lab that noted that there were 134 G protein coupled receptors that have served as targets for FDA-approved drugs. And you're welcome to look at those papers to see where the differences have arisen from. However, I'll point out to you in the graph that's shown that the 108 that they found in the Hauser paper, they've included then drug bank and IU-FAR receptors within the receptors as well, and that gives the 134 that they came up with for Inzel's paper. So it's clear that these receptors represent a huge market. They suggest that the global annual revenues for G protein coupled receptor targeted drugs are in the neighborhood of $180 billion. And in fact, that represents 27% of the global market share. So G protein coupled receptors, clearly a popular target in the past. And the question really is, what about going forward? So this is just a table that shows a few of those drugs from the 69 that have been approved in the last 5 years. And you would see, if you looked at them, that they cover a wide range of indications, everything from CNS disorders like schizophrenia and depression, insomnia, narcotic cough, right? You have a very wide range. There are some cancer indications for basal cell carcinoma with the smoothened antagonist that we have, and hyperparathyroidism with the calcium sensing receptors. So it's a very wide range that in terms of disease indications. But what's going to happen looking ahead? So I think that you would find that GPCRs are going to remain a very popular target. Currently, there are 321 drugs that are targeting GPCRs that are in clinical trials. Of those, nearly 20% are targeting novel GPCRs. That means that that receptor has not been targeted in the past. From those, many of them are orphan G protein coupled receptors. So as I mentioned earlier, an orphan receptor means that they don't know what normally activates that receptor endogenously in the body. But even without that knowledge, they're able to identify new drugs that are going to target that receptor, and they've been able to identify when activation or inactivation of that receptor would prove useful for a specific disease. So I've listed a few of the orphan G protein coupled receptors that are going to serve as targets currently within clinical trials. On the right in the table, I've indicated some of the receptors along with the compounds that are being used to target them that are all within clinical trials currently. Now, you would, if you did the math, from the 800 receptors, the 108 novel receptors that already have targets, and you subtract all that out, what you would find is that there are still, though, about 224 receptors that remain to be exploited. There is no drug yet, not even being tested in clinical trials, that would turn that receptor on or off. So, if we want to look at the breakdown for some of the drugs that are currently being tested, I would tell you that one of the areas that's really expanding has to do with biologics. So of the 800 G protein coupled receptors, about 118 of them recognize either peptides or proteins as their ligand, and about 30 of those already have an FDA-approved drug. Now, in some cases, those are small molecules, and in other cases, they're biologics that actually mimic the peptides that activate them with another peptide. And I've given you some of these examples for such as the GLP, the glucagon-like peptide receptor that has multiple peptides that are being used for it, somatostatin receptor, melanocortin receptor. All of these have either peptide or small molecule modulators of those receptors. I would say one of the reasons that you have been able to see this expansion in biologics is that many of these receptors, these peptide and protein receptors, have now been crystallized. So 21 of the 118 receptors have been crystallized. And again, I have provided a reference that nicely shows this variety of crystal structures that really helps us see what the receptors look like. I also point out here that of all the peptides that have entered the clinic since 2010, 40% of them target G protein coupled receptors. So while they represent 34% of all targeted drugs, they represent 40% of all the peptides. And I think that this dominance of the biologics in terms of peptides and proteins for receptors will continue. There are currently 16 G protein coupled receptor targeted biologics that are in clinical trials. So what about monoclonal antibodies? Oftentimes monoclonal antibodies are representative of the expansion of biologics, but that hasn't really happened as much in the case of G protein coupled receptors. So there is one antibody that is currently approved. However, it's only been approved in Japan. It is not yet approved here in the U.S., and it is a CCR4, a chemokine CCR4 antagonist. However, that being said, there are a number of clinical programs on G protein coupled receptor antibodies that are in progress. They represent 37 different receptor targets, and we have them at all three phases of clinical trials. So phase 1, phase 2, and phase 3 all have new targets that are G protein coupled receptors that monoclonal antibodies have been developed for. In fact, there's been a nanobody that was directed at a chemokine receptor, CXCR4, that was introduced. However, it was terminated. The trials were terminated after phase 1. And again, as a really good point of reference, there's a recent review in Nature Reviews Drug Discovery specifically about monoclonal antibodies that target G protein coupled receptors. So you might ask, what is fueling the hunt? Why this interest still in G protein coupled receptors? Well, I think without a doubt, one of the reasons has to do with the increasing number of structures. The second would be expanding pharmacology of G protein coupled receptors. There have been some novel approaches to screening that have been introduced in recent years. I think there's a recognition for some new disease indications for G protein coupled receptors. And finally, just the advancement of pharmacogenomics and the recognition that they play in GPCRs, I think is also fueling the hunt for new drugs. So I'm going to take each of these topics and try and go through them in a little more depth. So the first would be the increased number of crystal structures. So rhodopsin was the first G protein coupled receptor whose crystal structure was determined to high resolution back in 2002. Since that time, more than 46 distinct G protein coupled receptors have been solved, and that accounts for over 200 ligand receptor complexes. 11 of these structures have been crystallized in the active state. Normally what happens when you want to crystallize a G protein coupled receptor is you have to stabilize it. These receptors are incredibly flexible and able to move, and you have to stabilize them in a single conformation that allows then for the crystallization process to occur. Some of the ways that they do that are to make thermostabilizing mutations. So these are point mutations within the receptor's complex. Oftentimes they add a T4 lysosome fusion that's added at the N-terminus of the receptor, or they use nanobodies to help stabilize the receptor in a selective conformation. I've also pointed out within this slide several websites that are quite useful if you're interested in looking at G protein coupled receptor structures. There's a database that's provided by the Zhang Lab, as well as a PDB homepage where you can look up crystal structures. One of the biggest advancements in terms of crystal structure that came forward for us is the structure of the receptor bound to the G protein. So that occurred in 2011. And that really allowed us to see what the receptor looked like in its active conformation as it activated the G protein. And we could begin to understand the contacts that were important between the receptor and the G protein and how the signal was actually sent from where the ligand binds along the receptor into the cell. So the transmission of information from the outside of the cell to the inside of the cell. But perhaps over the last year, this use of cryo-electron microscopy has really advanced our knowledge in the structural information of GPCRs. And the advantage is that cryo-EM allows us to use wild-type receptors. We no longer have to modify them. There's no need for the T4 lysosome that has to be added. And in addition, they offer us the ability to look at the structures at a much higher resolution. So, for example, these are two crystal structures, cryo-EM structures, that were provided in 2017, one of them for the glucagon-like peptide receptor and the other one for the calcitonin receptor, both in complex with the G proteins. And these structures are at a much higher resolution than that was provided by X-ray crystallography. So we're now at 4 angstroms versus 20 to 30 angstroms that we had previously in terms of resolution. And by having this information, we not only gain information about the general context of signaling and how the receptors work, but we can begin to really expand our in silico information that we're able to gain through looking at these structures. So in silico drug discovery with G protein coupled receptors has really been hurtled forward, and you will see now that they've been able to harness the information that's been provided through all these crystal structures, and they can do virtual screening now to identify novel compounds. They can take old compounds and see where they bind. They can look at new compounds and see where they would bind. They can look and say what interactions might occur in terms of off-target, and they can do this virtually. One of the other advantages of having all of this information from crystallography is that we're able to look at allosteric modulation, which we're going to talk about in pharmacology. And it's allowed us to use molecular dynamic simulation as well. So we can literally simulate what's going to happen and how that ligand would actually bind to the receptor. So all of these in silico methods have really improved over the last decade. So expanding pharmacology. So we've recognized that the pharmacology that underlies G protein coupled receptors is important. And I think, right, we had a bit of a sort of limited picture of how that takes place, how the receptor would activate the G proteins, and the G proteins then go on to activate downstream signaling cascades. But over the last decade or so, I think that understanding has really blossomed. So we recognize now that many of these ligands, they actually activate more than one receptor, and that receptors usually activate more than one G protein, and that the receptor can signal intracellularly in both G protein dependent and independent manner. So they can couple to proteins such as beta-arrestin completely independently of the G proteins and signal through those proteins. We recognize now that there can be functional selectivity, so you can have a ligand that activates one pathway preferentially over another. There's constitutive activity, meaning that the receptor can interact with the G protein, and that can be completely independent of a ligand binding event, that there is already an activity between the two of those. Now, that may be with the G protein, and it may be with the arrestin. We now recognize that allosteric modulation, where you can alter how the endogenous ligand binds to the receptor. We recognize now that receptors can be activated from within internal cellular compartments. So the receptors may be in the mitochondria or at the nuclear membrane, and that they can be activated, and that those receptors can signal intracellularly. I think we certainly appreciate now more the fact that receptors will both homo and heterodimerize, and that there's crosstalk with other receptors, such as receptor tyrosine kinases. So all of these have really helped to expand how we look at how receptors signal. One of the ways in which this is often described now has to do with something called conformational barcodes. And it's the recognition that the receptors actually can be activated, right? They may be phosphorylated, in those different receptors, whether phosphorylated or unphosphorylated, they serve as a barcode for the receptor to interact with different downstream signals. So one barcode with phosphorylation at two different sites may lead to one specific pathway being activated, and a second receptor that may be phosphorylated at 4 sites may send the receptor signaling down through a different pathway. I also, and I give you a reference if you want to look into this further, this idea of conformational barcodes as serving the receptors in terms of how they signal. And what's really important to recognize is that it's really the conformation of the receptor that drives its function. So some of the insights that we've had for crystal structures have really helped us solidify our understanding of the orthosteric ligand binding pocket. This was a great paper by Monk in Current Opinion of Pharmacology in 2016 that shows some of the various ligands binding to the orthosteric binding pockets of the various receptors. And you can see that they go all the way from the outside to within the transmembrane helices in terms of where they actually bind, right? It's not just a single pocket, because depending on the receptor, the pocket is going to be different. The other pharmacology that's really, I would say, been of interest has to do with allosteric modulation. So this figure shows in a cartoon fashion the idea that you can have orthosteric binding, and then the allosteric binding can alter that binding of the orthosteric ligand, but it doesn't have to. The allosteric modulator can change the efficacy that drives the signal from orthosteric ligand binding to function, whether that's G protein binding or arrestin binding. And so it doesn't actually have to interfere or change, per se, the binding of the orthosteric ligand. It can just modulate its efficacy of how it sends its signal across. And then you can also have allosteric modulators that just directly modulate how well the effector proteins, be it G proteins or beta-arrestin proteins, how those can bind to the receptor. And what we've clearly found is that you can find compounds that will be both a positive modulator, for example, of agonist binding, but a negative modulator of agonist signaling, or vice versa. So again, this is a paper. This one is from Vaidehi in 2016, showing, in fact, some of the diverse binding sites of allosteric modulators. And so you can see within this figure that you have ligands that bind to the extracellular portion or to the transmembrane domains, as well as ligands that bind intracellularly that are near the surface with the G protein, for example. And they really litter, if you will, the receptor from the outside in terms of these allosteric pockets that are available. We actually have 2 FDA-approved allosteric modulators. So cinacalcet, which is a positive allosteric modulator that was introduced by Amgen for the calcium sensing receptor, and then maraviroc, which is from Pfizer, that is a negative allosteric modulator of the chemokine receptor, CCR5. So clearly, allosteric modulators are something that are going to be in the future. And just to give an example of this, you can see that we have many allosteric modulators now that are in the clinic for a wide variety of G protein coupled receptors. So this is just a few of them. This is not an extensive list, but it clearly shows the variety of receptors that now have been targeted by allosteric modulators. So one of the tools that is available to anyone who looks at G protein coupled receptors is the GPCR database. So this database, and I give the site for it, actually has some 144,000 ligands that allow you to look at a receptor and its interaction with that ligand, and then it captures the effect, either qualitative or quantitative, and then any mutations that have been done to that receptor, and then measure how that ligand still binds, what its potency is, what its affinity is. And it's a phenomenal database that anybody is able to use. There are some other databases that relate to GPCR ligands, and I've listed just a few of those, again, on this slide for you. So I mentioned functional selectivity before, also known as stimulus bias signaling. It is something that is certainly more recognized now than it was 10 years ago. And it's the idea that a ligand can stabilize one conformation versus another conformation when it interacts at a receptor. So, for example, if ligand 1 interacts at the receptor on the left-hand side, you may get signaling through GI, leading to a decrease of cyclic AMP. But if you have a different ligand and it binds to the exact same receptor, you may get signaling through GQ and an enhanced calcium response. So the same receptor leading to two different responses based on what the activating ligand is. But bias doesn't have to be just with G proteins. It can actually be between G proteins and beta-arrestin, right? You can have selectivity of activation of a beta-arrestin pathway leading to things like receptor internalization or MAP kinase activation. So I give you an example of this. This is the apelin peptide or a drug, CMF-019. And in this case, they've looked at three different responses. They've looked at cyclic AMP inhibition, they've looked at beta-arrestin activation, or they looked at receptor internalization. And what you see is if you look at these and you compare them one to another, that the CMF is actually a biased agonist for the apelin receptor. And so it shifts the signaling to the G protein pathway versus beta-arrestin or internalization. And so it's not nearly as good at either of those, whereas the apelin 13 is equipotent for all three of those. So you might ask, is there any FDA-approved biased modulator of a G protein coupled receptor? And I would say yes. However, the one that we know of is carvedilol, and that is a beta blocker. And the beta blocker that has some ERK agonist activity. And what they found is that that drug has reduced mortality when compared to other beta blockers. But they don't know yet if the difference in the therapeutic efficacy is actually related to its bias or not. And so that's currently being studied. There's been some nice reviews that have just recently come out on that, and I think that this is something that will be explored. It's important to note that when this drug actually came about, functional selectivity, the appreciation of that, hadn't even been described yet, and so it wasn't being tested. So multiple pathways were not being tested as part of a routine drug screening program at that time. They didn't look to see if the drugs were identical in how they signaled the receptor down GI versus GQ versus GS, right? They just looked at a single response. So I would say, again, that the pharmacology has helped drive the screening and vice versa. So we have a variety of new tools at our disposal. These include things like high content screening and ultra high throughput screening capacities. The use of, for example, the delta log, where you look at both the Emax and the EC50 for the compounds, so that you can quantify whether your ligands are biased or not. We have a number of non-radioactive methods for things like IP3 accumulation. We have real-time assessment now of cyclic AMP. We have cell surface biosensors and Brett assays that really help determine things like receptor internalization. There's been a big trend in optogenic and DREAD studies that allow you to use receptors that only respond to a certain stimulus. There's been a number of new in vivo studies that show, for example, some fancy fluorescent engineered reporters that allow us to look at signaling of receptors, these G protein coupled receptors. And then finally, as already mentioned, right, this availability of GPCR crystal structures has really helped us in our computational campaigns as we screen for novel G protein coupled receptor drugs. I think one of the clear inferences from all of this information is that there's been an expansion in the indications where GPCRs are recognized. And that's impressive, given that they already had such a wide range of indications, right? We have drugs for things like asthma and cardiovascular disease that all target these G protein coupled receptors. But we're moving now into areas that didn't have much representation before. So one of those is cancer. I actually show a graph that showed that less than 10% of cancer targets were cell membrane associated. So the number of GPCRs in the field of cancer drugs is pretty small, but that is expanding. So we have recognized now several new receptors, and they're placed in a variety of tumor types. The other expansion has been within CNS diseases, where we've recognized receptors that we've known about in BlueR5, for example, but we've now been able to selectively target them, and by doing so have potential new drugs for what are well-known diseases. I will point out that this success has not come without failures. There's a report from Genetic Engineering News that talks about the top clinical failures for 2017, and 3 of the 13 happen to be G protein coupled receptors. I'd say that the other area that's sort of just now coming out of the dark has to do with pharmacogenomics in the context of G protein coupled receptors. So pharmacogenomics relates to how genetic variations can alter a person's response to medications. And typically when we think about pharmacogenomics, we think about it as either altering the pharmacokinetics, that's things like drug metabolism or transport, or the pharmacodynamics, which is the target itself. And so for the context of G protein coupled receptors, I'm talking about pharmacodynamics. What is it about the pharmacogenomics of the target itself? And currently, no receptor variants are included in any of the labeling information for the 475 GPCR targeted drugs. But a recent paper, which I've provided for you, a Cell paper, actually looked at information from the 108 GPCRs that have drugs, and they found from genetic information that there was an average of 4 common and 128 rare variations within that lot. And they actually took that a step further, and they analyzed the impact of just 3 variants that happen to occur within the ligand binding pocket of the mu-opioid receptor, and how those altered the binding of morphine, buprenorphine, or naloxone. And they found that there were distinct differences with either loss of function or gain of function for those mutations with those specific ligands. So in summary, I would say that despite the fact that G protein-coupled receptors are the most successful drug target in history, that they haven't been fully exploited as a target for the treatment of human disease. As I said, we have 224 receptors that still haven't been moved forward in the context of disease. We've made substantial progress in our understanding of the receptors, what their structures look like, and how they in fact signal, their molecular pharmacology. There's a lot of new tools that are available to us that help us understand the binding between orthosteric and allosteric ligands, and functional bias. The application of some of these structure-based drug design, right, in silico ligand docking, the molecular dynamic simulations, they've all been made possible because of this expansion of GPCR crystal structures. And I think we are right at the beginning at this idea that genetic variations that occur in functional sites on G protein-coupled receptors may result in altered drug responses and may ultimately underlie some of the pharmacogenomic differences that people have in how they respond to GPCR-targeted drugs. And with that, I will actually answer some questions if you have any.
Thank you so much, Dr. Gilchrist, for that informative presentation and also for your important research. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question drop-down box located on the far left of your presentation window. Type that question into the box that appears on the screen and click the Send button. We'll answer as many questions as we have time for in today's presentation. So let's take a look at our incoming questions from our current audience members. Our first question, Dr. Gilchrist, is why have clinical relevant monoclonal antibodies been so slow to emerge for GPCRs?
Question. I think that, right, because there's only one that's currently made it all the way through the clinic, and that's only been in Japan. But there are over 50 monoclonal antibodies that are out there that have been FDA approved. Someone certainly would ask why haven't GPCRs been better represented, and I think it has to do with a couple of different things. I think one of the problems with G protein-coupled receptors was the same problem that we had in determining crystal structures, and it's that they have such conformational variability. They can change their shape and they're able to be incredibly flexible. And so getting them to lock into a specific conformation that's therapeutically relevant and that be the only structure that the antibody sees and responds to just has to do with making those antibodies has been very difficult to get that conformationally constrained receptor and to know what that should look like. So I would say that's definitely been a problem. But I think that it's one that they are grappling with and that they've have found some answers to and that's improving.
Wonderful. Thank you, Dr. Gilchrist. And thank you to our audience members for their participation today. And I want to remind everyone that any questions that are not answered in today's presentation will be answered and addressed via email. Our second question coming in is, given the recent failures mentioned, is there a way to mitigate the risk with GPCR drug discovery?
Another answer. Certainly every pharma company would love to know if they could mitigate their risk. I would certainly think that by having some of the bioinformatic and chemoinformatic information that's now available, it's going to be much easier to screen in vitro through virtual screening to determine whether your ligands are going to have any interactions at other receptors. And so that's probably going to help, and I would really think that using that information as broadly as you can will help mitigate the risk going forward.
Thank you very much. And do you have any closing remarks that you'd like to provide to our attendees today before we close?
I would certainly say you're going to have to keep up on the reading. GPCR is an ever-changing field. I've done my best to give you some great websites and some resources and some papers that are all quite helpful in terms of where GPCR research is today. But as the field changes, you have to really keep up with it. So stay in tuned.
Thank you again, Dr. Gilchrist, for your presentation and for the work you do. I'd also like to thank today's LabRoots for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through May 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thanks for joining us. We hope to see you again soon. Bye-bye.